Welcome to the Infection Prevention Strategy Tips Masterclass webinar, Continuously Active Disinfectants, Addressing Challenges of Surface Contamination. We will get started in just about a minute. Welcome to the Infection Prevention Strategy Tips Masterclass webinar, Continuously Active Disinfectants, Addressing Challenges of Surface Contamination. My name is Dan Mueller. I'm the moderator of today's webinar. I'm a vice president at Garcia Medical and also a TIPS advisor. Listed below are the learning objectives. After completing this webinar, you should be able to address the following points. You want to read those? By participating in this webinar, you acknowledge that the statements and information provided is for general guidance purposes only. You are advised to contact your local government and regional health authorities for specific instruction on all the topics mentioned in this webinar. And this webinar is not to replace the guidance and direction of your local government and health authority. The webinar is hosted by TIPS. Our free services include Science to Market program, the TIPS Journal, deep dive interviews, and masterclasses. www.ic.tips. The mission statement is TIPS is the following: a non-for-profit organization that exists to advance innovations, ideas, and processes that make a difference in global health. TIPS extends over 30 countries and is a strategic partner for many notable organizations and initiatives and events. The motto of TIPS is join, contribute, make a difference. If you have any questions during the webinar, we will hold all the questions to the end. You see the Q&A icon. You can type in a question and we will get to as many questions as possible. If we run out of time, we do record the questions and we will follow up with you after. For today, we have a sponsor, which is EverClean. Gabriella, are you on the line?
Yes, I am. Thanks, Dan. We are the EverClean Infection Prevention Solution by EarthSave. While you can find sprayers online and you can find chemicals online, we really pride ourselves on being the entire solution. We have a simplified, standardized approach that focuses heavily on safety, efficiency, and cost savings. Our system provides targeted education and training across every industry. Our simplified approach is an easy two-step process. Step 1 is our Pure 1, which is a cleaner disinfectant. Our Pure 1 is the first product with a registered kill claim against bacteria and biofilm. Pure 1 can be used with our dry wipe system on all surfaces, as well as with the mop and bucket for the floors. Our new environmental surface wipes have been proven to mitigate cross-contamination and clot binding in the hospital setting. Step 2 is our enhanced touchless disinfection. Our Protexis sprayers emit a positive charge, which creates a magnetic effect to all surfaces, gaining 360-degree coverage in half the time of traditional methods. At the higher dilution, our chemistry has a 4-minute C. diff kill claim. For more information on the EverClean Infection Prevention Solution, please feel free to reach out to Rich Prinz or myself.
Thank you, Gabriella. I know I've heard some of the clinicians are in serious need of CE credits without any in-live meetings. So if you're looking for a CE credit, we are offering 1 CE for nurses. After the webinar, you'll receive an email, and if you're interested in the CE, you'll need to fill out a survey to receive the 1 CE credit. We will also be recording the webinar. If you know anyone that was not able to attend, we will send more information out through the TIPS network. Without further delay, I want to introduce today's speaker, Dr. Rodney Rohde. Dr. Rodney is Professor, Research Dean, and Chair of the Clinical Laboratory Science Program in the College of Health Professionals of Texas State University. He has been recognized with teaching excellence at both Texas State and Austin Community College. His background is in public health, clinical microbiology, and his PhD dissertation at Texas State included MRSA knowledge, learning, and adaptation. Rodney has received numerous awards, including a top 20 professor at CLS, the Texas Mentoring Award, Distinguished Author Award, and ASCLS Scientific Research Award. Dr. Rohde is also an advisory board of TIPS. Our second speaker is Dr. Kelly Reynolds. Dr. Reynolds is professional and chair of the Department of Community Environment and Policy, program director of Environmental Health Sciences at the Zuckerman College of Public Health, and director of the Environment, Exposure Science, and Risk Assessment Center at the University of Arizona. She has spent over 30 years of experience in academia, specializing in tracking pathogen movement in healthcare environments and evolving predictive risk assessment models for determining pathogen spread, human exposure potential, adverse health impacts, and intervention efficacy. Dr. Reynolds has served as primary investigator on numerous projects and published over 375 journal articles, book chapters, and professional reports. Dr. Reynolds is also an advisory board member of TIPS. I will now turn it over to Rodney.
Good morning, everyone. Thank you for joining us today for this important webinar. And just want to start off here with talking about really what everybody's been talking about for the past 15 months, and that is invisible pathogens. So not only are we always concerned about something like SARS-CoV-2, but we must not forget that there are many other bacterial, fungal, parasites, and other viral pathogens that are virtually everywhere. So they're very ubiquitous, not only in nature, but also in our healthcare and community settings. And we must remember that it's not just a hospital or healthcare problem. These pathogens are found in almost every niche and every surface that you look at, minus sterilized areas. And even there, they can show up again very quickly. So one of the questions that you see circulating right now globally with respect to the pandemic is, should we still be focusing on surface disinfection with COVID? And one of my favorite statements, both online and publicly when I speak, is that all surfaces matter to everyone at all times. So while we do know now, and most research is showing that SARS-CoV-2, which causes the COVID infection, is more likely to be spread through aerosols and the air transmission route, certainly viruses still are found on surfaces, just like other pathogens. It's really about understanding their persistence and the time they're going to be on those surfaces and how that interacts with human contact and how that may add to the chain of transmission. So that's kind of where we're heading today. So we know, if you observe this slide, that pathogens do have a lifespan on different surfaces. My esteemed colleague, Dr. Reynolds, will be talking about some of this later in her area, but just generally, when you look at some of these pathogens that many of you may be aware of, like methicillin-resistant Staph aureus and just general staphylococci, look at the time that we've seen these things persist on surfaces: 9 months, 7 months. Of course, when you look at Clostridium difficile, we know that the vegetative pathogen can last for a while, but the spores, the endospores that C. diff produces, can last for even longer amounts of time. And then different viruses, norovirus and SARS and HIV and so forth, can last certainly days and weeks. And that kind of depends on whether or not they are enveloped or non-enveloped, which we'll kind of talk about with this slide. So this is a general slide. It's a pretty famous slide. You can see this in various formats with different talks. And it really just shows you a ranking or a hierarchy of the different pathogens with respect to how they are resistant or sensitive. So on your left, you see the arrow at the bottom. The S stands for sensitive, meaning they're more susceptible to agents. And then as you go up the table, things become more resistant. So if you start at the bottom, and we won't go through every line here, but the bottom, thankfully, when you look at things like SARS, it's actually very sensitive to different types of biocides and different types of disinfectants. So enveloped viruses tend to be that way because the outer surface is more lipid, more fat-based, and they're easily penetrated by different things. So they have a very low shelf life on a surface. But as you start going up that scale, you can see we move through the gram-positive bacteria, the larger viruses, which are non-enveloped. Non-enveloped viruses tend to be more resistant because they don't have that lipidy envelope. And then you move into some of the gram-negative problematic bacteria like CRE, which many of you know about, the carbon-resistant, carbapenem-resistant enterobacteria, Pseudomonas, Acinetobacter, and some of the others. And then you move into the fungal spores. Right now, I've just completed an article about Candida auris becoming even more resistant. In some areas, it's resistant up to 3 of the latest antifungal drugs. So that's a problem, especially for immunocompromised populations. And then you move into other small non-enveloped viruses, the mycobacterium such as tuberculosis, and then ultimately the spore-producing bacteria like anthrax and C. difficile are very problematic unless you use sporicides. We know that the World Health Organization, the Director General, some of these quotes you may have seen recently, but certainly we know that the emergence and spread of drug-resistant pathogens, and that crosses all areas of microbes, has accelerated. More and more essential therapies and medicines are failing. In fact, we see that the therapeutic arsenal is shrinking, and the speed with which those drugs are being lost actually outpaces the development of replacement drugs. So according to WHO director, even the research and development pipeline for these new antimicrobials is basically running dry. And this is due to a lot of complex factors, but it is unfortunately upon us. So really and truly, disinfection and prevention is the last line of defense. And the famous saying that an ounce of prevention is worth a pound of cure is something that I continually speak about when people talk about antibiotics or antivirals or even vaccines. I mean, truly preventing the infection is one of the best ways to avoid dangerous pathogen transmission. We also need to be aware of how infected populations versus colonized populations can contribute to this issue. In general, when I speak to the public, most people understand this, that when you have an infected individual, an actual infection, you can sometimes certainly identify that person through testing, through symptoms, through how they look, syndromic issues. Those are what I would call the more easily identifiable issues. But when you understand that there is a under the surface colonized population that you don't easily identify, and you may not even be thinking about testing for, then you have kind of this hidden population that can add to the transmission chain of infection, including surfaces from people that don't realize they're colonized. When I first started out working with MRSA, one of the things that I quickly learned through the literature and then through testing and within my own populations is that if I were to swab everyone that's on this webinar, your noses today, 30% of you would be colonized with regular old Staphylococcus aureus. I've done this numerous times in different populations, including healthy populations, prison populations, dorm room populations, physical therapy professionals, and the list goes on, nursing, and it's right at 28 to 31 or 32%. So a third of the population is often colonized with regular Staph aureus. And then if you look at MRSA, that's usually a little less. It's down in the 1 to 2% colonization, but certainly you have the potential to spread those different types of bacteria and other agents, even when you don't know you're infected, or when you don't know you're colonized, excuse me. So this is another quote coming out of the Centers for Disease Control from a physician stating that the threat of untreatable infections is very real. Obviously most of us back in the 60s, 70s, and maybe even 80s thought that this was unthinkable, where antibiotics don't work. But it is upon us. We're already seeing this. Many of you know this. Germs are becoming much more resistant and persistent than any antibiotics we have to treat them. This is a very real thing. I've experienced this in my own family. My father has an MRSA infection that continues to persist, and it's one of the reasons I'm so passionate about this topic. I continue to call this, and I actually wrote about this in a recent TIPS publication, that we are dealing with a slow burning pandemic, and that's called antimicrobial resistance. And Dr. Tom Frieden, the former director of the U.S. Centers for Disease Control and Prevention, has stated numerous times that we risk being a post-antibiotic in a post-antibiotic world. I would go one step further in saying that we are there. We are at the doorstep of the post-antibiotic world with respect to some of these agents, not completely across the board for sure, but we are seeing some agents now that just are almost pan-resistant to all antimicrobials, and that's a scary thing, meaning that we really need to think about cleaning and disinfection procedures to be more essential to mitigate those infections. These types of bugs are often referred to in the general press as superbugs. Some people don't like that word, but I think it does a nice job of kind of talking about these pathogens that are almost on steroids, right? They've developed the ability to outlast and maneuver around antibiotics and antimicrobials. And it's not just bacteria. We're seeing this with fungal agents as well as viral agents. So we truly are the last line of defense when we look at infection prevention and cleaning and disinfection. Not to go too deep into this infographic, but it's a nice infographic showing how antibiotic resistance happens. In a nutshell, when you take a human body or an animal or an environmental source like a body of water and you treat it with an antibiotic, especially if you treat it in a uncontrollable way, over time, what happens is that those bacteria and other microbial normal flora that you're supposed to have will be killed off, and those bugs will select for the more resistant bacteria. And so as you get to number 3 and 4 there on the top of the graphic, you end up getting a more normal drug-resistant bacteria floral, as well as now you have bacteria that can share their resistance factors with other bacteria through various mechanisms that I'll mention soon. And then when you drop that into the kind of the global One Health paradigm, you start seeing that animal health and environmental health interacting with humans can spread that antibiotic resistance organism through patients, through healthcare professionals, through animals, through their feed and through their contributing their waste to the environment, as well as to the surfaces within the community and healthcare setting. And so all those errors, what's scary about that diagram is that there's never really a stop point, right? It's a continuous cycle. And unless we find ways to remove those pathogens from those environments, we're going to continue to have this problem. Here's a quick diagram that just kind of shows you that bottleneck effect. Recently published an article on this with a colleague, and this is really a general known phenomenon in antimicrobial world, is at the top of the funnel you see a variety of normal bacteria, different normal flora. Some of these may even be good and healthy for you. And when you start treating with any agent, what happens is you're going to kill off some of those different colors there that are normal and good bacteria, and you may not ultimately get rid of the more resistant green looking organisms, and then they become the primary population, leading to the rise of an antimicrobial resistant pathogen. We also know that weak biocides can increase bacteria's ability to combat antibiotics. So these are used in the food industry, often at sublethal doses, and that means that they can end up endangering rather than protecting the public health, because again, we're increasing resistance in these different microbial bacteria, and we're enhancing their ability to form biofilms. Biofilms typically are seen in 60% of human infection. Coming out of this biofilm formation, primarily in wounds, although it can be in other areas. Biofilms are this kind of sticky matrix of material that acts as a reservoir of pathogens in the hospital and in the community, and they kind of offer these nice protective environments to flourish. It protects them because they're embedded and biocides often can't get to them. And they basically increase patient exposure and provide a very high infectious dose in those types of systems. Most of the time we're talking about at-risk patients being immunocompromised patients in healthcare. It can be other situations, but these are the general higher risk. Cystic fibrosis patients, anyone that's on a breathing machine or any other type of mechanical ventilator, as you have seen in the pandemic, catheters or anything that's going into the body that's giving a highway to a pathogen, different wounds like surgery and burn victims, preemies and other types of cancer patients. UTIs are problematic because of catheters, and then prolonged stays in hospitals. Those are all really serious risk factors for biofilms. Again, biofilms are an interaction, often of different bacteria. There's a higher density of cells that can lead to something called quorum sensing. And you can think of quorum sensing as a way for a way for bacteria to communicate within an environment, within that biofilm. They literally are sharing their defenses, their growth factors, even their very DNA. And so they're sharing their ability to become resistant. This is a diabolical, problematic trait that you see with these different bacteria. It's really problematic because they're basically sharing secrets with each other when we don't always know it. The older these biofilms get, you can almost think of it as being both a living biofilm, but also a hibernation area. So if you start hammering them with biocides, they can actually go into hibernation, which makes it harder to kill. And then that can lead to something we call persister cells, meaning they're persistent, they persevere, and those become more difficult to kill by either antibiotics or disinfectants. And so when they're threatened, they shut down. They're very smart, and then they start repopulating the full biofilm with a very high density of these persister cells after the antibiotic or disinfectant threat passes. And that learned immunity, that shared learned immunity in biofilms, is then released in seeding phases, which can include large pathogenic globules of biofilm. You can almost think of these as, an army that's able to teach their best defense mechanisms and attack mechanisms, and then they kind of send out these little units, these patrols of bacteria out into other areas that are naive and set up shop there. So it's really an effective way for these resistant bacteria to operate. Very diabolical. And unfortunately, they're found everywhere. Moist areas with nutrients and surfaces to attach on. And so kind of a very basic idea is that you have this kind of transient attachment of bacteria. They become stronger, adhered to the surface. You start getting this matrix and aggregation of cells with different nutrients and kind of this slimy layer, if you will, of different types of filming and nutrients. And there they grow and mature. And then they start doing this piece I talked about where they'll start dispersing globules out from that primary surface. So the diagram here, the infographic kind of shows you how bacteria start out as a planktonic form, which they're growing kind of more in a liquid type of setting, and then eventually they like to attach. At first they can reversible, so they can kind of attach and detach. Eventually they become more reversible, and that biofilm forms, and that's that dangerous community where they can share through quorum sensing as they grow and mature. Just a nasty, nasty defensive mechanism. So you can see some of the different impacts, everything from water coolers, food processing, ships and implants. Certainly medical implants are problematic. Biofilms grow in your teeth. That's why you brush and floss to get rid of them. In other areas, anything with a surface can be a compatible site for these biofilms. Again, just kind of showing you visually, not only in healthcare, but it can also show up in the environment, all sorts of places. One recent article that I talked about, which really is kind of a scary thing if you read about these, are microplastics. And not to read this to you, but microplastics are very small. That's not 5 meter, that should be 5 millimeter, very tiny. fine plastic particles, and they're found in all kinds of things: cosmetics, your toothpaste, your clothing, everything. They're found everywhere. And it shows that average size wastewater treatments that can serve up to 4,000 or 500,000 residents will discharge up to about 2 million of these particles into the environment every single day. And a recent Journal of Hazardous Materials commentary talked about researchers that found certain strains of bacteria that were very resistant up to 30 times while living on these little biofilms on microplastics. They kind of formed a activated sludge, if you will, at these wastewater treatment plants. And so they basically become hot spots, because you can imagine these biofilms forming on these plastics. They're sharing their information through quorum sensing, and they're moving about in that way. So again, very problematic, because certain bacteria will attach. They will secrete these glue-like extracellular substances that we know as biofilms. That allows other bacteria to stick there and grow, and then it gives them the opportunity to exchange DNA. And again, researchers are worried about this because this spread of antibiotic resistance genes now is becoming more common in the community. So we're going to have to really keep an eye on just water and wastewater treatment, as well as other areas. Ultimately, to wrap up, biofilms provide a protective form of growth from environmental stress. It's about 1,500 times less susceptible, 1,500 times less susceptible to certain biocides. And this diagram shows you that interprotection there really protects it from all sorts of things, not just biocides, but also from phagocytosis of the human immune system, UV light, dehydration, and other types of attacks that you might try to kill organisms with, while providing access to nutrients and opportunities to interact among that biofilm. Just a really ingenious and diabolical system for these bacteria to function. Just another quick infographic to kind of show you how these things are living on the surface, both in anaerobic and aerobic formats, and how they have substrate and oxygen available to some of those formats, and they persist in these areas of attachment. One quick comment, if you're wondering how these bacteria share these resistance genes, if you're familiar with some of the basic microbiology of bacteria, they do something where they share extracellular DNA. So these are little tiny chromosomes. If you look in the picture to the right, and you see these little yellow circular components called plasmids, those actually can have resistance genes. So little segments of DNA that make it resistant, for example, to methicillin or to some other antibiotic. It also may have a gene to show resistance to a biocide, so whatever it is, or artificial growth factors and things like that. And they can share those through a process called transformation, conjugation, which is where bacteria utilize these little tunnels. To the right of that diagram, two bacteria that have pili. These are little things that touch, and they literally let bacteria slip that plasmid into a naive bacteria. So the red plasmid can be shared into the other bacteria that doesn't have it yet, and now it's picked up all of the characteristics of this resistant bacteria. I often talk about it. This would be equal if a human being could touch fingers with another person and share their blue eyes, right? So I mean, it's sharing traits. You don't have to reproduce, you just touch and share. This is really another rapid way to share resistance. And then transduction is also a way through bacterial viruses where they inject their DNA. So just, again, diabolical ways for bacteria to pick up these resistance genes and create these problems within biofilms. I believe that is my stopping point, other than this last slide on persister cells. So when you look at that lowest level on that surface, these persister cells, again, are kind of these persistent colonies and communities of bacteria that form over time, and they end up being less tolerant. They are specialized in how they survive, and it really allows that biofilm to regenerate quickly, and unfortunately regenerate with a higher resistant nature, because they have already become resistant to those biocides as well as antibiotics and things like that. think of them as being embedded at that surface in that slime layer. And it's very difficult to get drugs and biocides into that slime layer. So it's very problematic once these develop. Now we'll transition to my colleague, Dr. Kelly Reynolds.
Rodney, fantastic presentation, very interesting.
Thanks, Dan.
Thank you, Dr. Rohde. This is Kelly Reynolds talking. I think we're all a little bit more frightened right now about how much smarter bacteria are getting, but I bring good news because bacteria are not the only things getting smarter, we are too. And obviously this session is about addressing these challenges of surface contamination. And I'd like to move our session into talking about some of the research around continuously active disinfectants. So some of the research that has been published recently has really come out of primarily Karen Vickery's group and collaborators that have been working with her. She really had this landmark study in 2012 about the presence of biofilm containing viable multi-resistant organisms despite terminal cleaning in clinical environments. And in particular, they looked at intensive care units. So when we look at the capability of surface biofilms to have an increased tolerance to biocides, one of the things that they looked at in the study was even after 2 applications of bleach at a really high concentration, 10,000 parts per million, which is about double what the CDC guideline is for surface disinfection in healthcare environments, even after 2 applications, the biofilm still was not eliminated. And we know that biofilms, as Rodney mentioned in his talk, are highly resistant to other biocides, including UV, and as we're talking about with bleach here. But other oxidizing agents are generally more effective against biofilms compared, well, oxidizing agents are generally more effective against biofilms than other chemicals that might also be prevalent, used commonly in the healthcare environment, like alcohols, phenols, and quaternary ammonium compounds. So the key message here really is that if your disinfectant is not working against biofilms, you're really not achieving adequate disinfection. We know that organisms that are so embedded in biofilms that our methods for monitoring how effective our cleaning is, for example, swabbing or ATP analysis, this sampling does not necessarily detect what's still present in the biofilms, these persister cells, as Dr. Rohde was mentioning. So in some of these studies, you'll see that to really release the biofilm from the surface, they had to do what we call destructive sampling, taking pieces of furniture and fabric and other types of surfaces back to the laboratory and really scrubbing and eluting these biofilms off. So you might think, well, if the biofilms are that hard to get off of the surfaces, then how are they coming off to cause infection in people? But what happens over time is these dry biofilms, as we call them, absorb moisture from the air in high humid environments and just in microclimates from shower aerosols and things like that that are normal to our environment, and they actually promote the growth of the biofilm. So looking more specifically at some of these studies that I mentioned in the first slide there, breaking it down, this is showing examples of the tolerance of dry biofilms to sodium hypochlorite or bleach. And you're looking here at a visual of, it's a viability stain, we call it a backlight staining. And in green, you see live cells. So on the left of this, you see a Staph aureus biofilm that was untreated. So a healthy population of the bacteria growing in a biofilm environment. And then on the right side, the B side, you see a treatment with sodium hypochlorite, and this was up to 20,000 parts per million for 10 minutes. They looked at a range of treatment from 1,000 parts per million to 20,000 and looked at what happened with the biofilm population. And at the highest best case scenario concentration, you see a lot of the green cells are gone, that the viable cells are now dead, but you still see a healthy residual of cells. And this is what we're talking about with these persister cells that can then regrow and recover in these biofilms. So more about dry surface biofilms, and this is from the original paper by Karen Vickery in 2012. So we've known about these for quite a while, but not really understanding completely on how to mitigate these events. So in her original study, she found evidence of these biofilms in the ICU on blind cords, so more like soft surfaces, fabrics, on door surfaces, reagent boxes, curtains. And, you know, really, I think Rodney detailed very well, I won't go into much detail about it, is the EPS, or that extracellular polymeric substance that really protects the bacteria and embeds them in the biofilm. And so you're seeing some evidence here. The vertical arrows in the figure on the left are showing the actual colonies of the bacteria. And then the horizontal arrows are showing you evidence of that EPS gelatinous material that is protective of that bacteria and allows it to persist. So in another study, also a co-author with Karen Vickery, this is Hu et al. In 2015, found a high prevalence of dry surface biofilms on multiple surfaces in an ICU unit. So 93%, that should be 43 out of 44 surfaces, were positive for a dry surface biofilm, and 52% of those had evidence of MDROs. So some of the surfaces that they were targeting were both soft and hard surfaces in these environments where the biofilms were persisting. And again, the biofilms were still present after 2 rounds of terminal cleaning with a 500 parts per million free chlorine solution. So Hu et al. Took it a step further too and looked at exactly what species are present in these biofilms, because we all hear about the good bacteria, you know, not really causing infections. And the reality is that in a healthcare environment, when you have immunocompromised patients with wounds that are exposed and new entry routes for bacterial colonization, any bacteria has the ability to colonize and create problems. So they're all somewhat opportunistic, but some obviously are more opportunistic and some are just directly pathogenic to the human host. So what Hu found was that there were multiple species in these biofilms, and you can see a list here of some of the most common species. And some of these are just inherent bacteria to our environment, but some of them are frank pathogens like Staph aureus, and some are more opportunistic like the Pseudomonas species. But overall, what they also found was when they stored these surfaces where they found biofilm for up to a year in dry storage, they could still culture MRSA, VRE, and ESBL gram-negative bacilli. So these are highly, highly persistent bacteria. So this creates obviously an implication for infection control and a new target for achieving a higher level of disinfection because of the prevalence of these dry surface biofilms and the knowledge that most cleaning procedures that we are currently using are not completely sufficient to remove or kill these dry surface biofilms. Now, these products can do a great job at reducing the concentration of planktonic bacteria, these kind of free-floating bacteria. You can show efficacy of 6 and higher log reduction, but we have never really tested routinely for these persistent cells in the dry surface biofilm and their ability to regrow. And we really think that this is a root cause of the endemic HAIs in healthcare environments. So I'm trying to see if there's anything else here I didn't cover. I think that's it. We can move on here. So just to summarize, this is the target, our pathogens living in the mainly in the biofilms, and their ability to survive. Consistent with Dr. Rohde's presentation, these biofilm persistent pathogens can live up to 15 or can be up to 1,500 times more resistant to our disinfectant protocols, and they regrow very quickly. Bleach and even hydrogen peroxide, which is another typically effective oxidizer on planktonic cells, are not completely effective. And we need to look at new technologies to address these biofilms. So how can we target it? Now, detergent can be very effective to break down that EPS layer in the biofilm. So cleaning can be very important, not just for removing organic matter so that disinfectants can work more effectively, but also breaking down the EPS in the biofilms. But we also know that detergent alone is not sufficient to really target the EPS, the biofilm, and the pathogenic microbes that might still be present. So targeting these, we think we really need to look at a new standard for disinfectant efficacy. And this is being talked about a lot by the EPA. They have soft guidelines right now for evaluating product claims for biofilm, addressing biofilms. These are just guidelines. They are not regulatory standards, but you can see some of the examples of how, myself as a researcher has some guidance on how to set up experiments to really prepare surfaces with biofilm, with pathogens embedded, and to look at efficacy of disinfectants. And so we're going to be talking about some of the testing that we've done looking at this. And also new chemistries. We're hearing a lot about these continuously active disinfectants, these CADs. And what I'm going to focus on is some recent research on sodium dichloroisocyanurate. That's a mouthful. Let's just call it NADCC for short. And the data that we have, and this product does have an EPA claim to kill biofilm. So NADCC essentially releases hypochlorous acid. What we looked at was fast dissolving effervescent tablets. So really easy to store, easy to create and dilute yourself and dissolve in water and have a readily active disinfectant. So the resulting solution is incredibly flexible. It's a broad spectrum disinfectant, and I'll show you some more evidence of that. But here's a great thing too, is the pH is around 6 to 7, so less corrosive than your typical bleach product, and safer in terms of handling, easier for storage, safer for personnel. And the evidence is showing that it's actually more effective than your bleach product, a lot more stable and less prone to variability under different conditions of temperature and pH. So this HOCl molecule is really a simple uncharged molecule. So that's a key difference from the bleach molecule as well. And it does attack indiscriminately multiple sites on the bacterial cell and the virus layer. So the proteins, DNA, RNA, and lipids, it can attack all of those areas. And it's also effective against a wide range of pathogen types: bacteria, viruses, protozoa, fungi, and yeast. Acts quickly and more efficiently than bleach, as I mentioned before. It's more stable, and we have not seen any evidence of resistance to HOCl. Now, I recently published a paper with infection control tips, IC tips, looking at multiple laboratory evaluation of this product, and really a review article of third-party data in the United States where we've evaluated this product, and primarily looking at both surface antimicrobial efficacy, but also biofilm inhibition. And all of these studies consistently looked at a 24-hour residual action. So you apply the product, let it dry, in 24 hours, evaluate what its impact is on either biofilm regrowth or recontamination of surfaces. Are two key targets for reducing exposure to HAI causing pathogens. So the data shows the benefits of NADCC chemistry for the reduction of these surface bacteria, and including inherently disinfectant resistant C. difficile spores and the inhibition of biofilm growth. And we see this as a major discovery in the control of environmental contamination and infection spread. I think this is the next layer of protection that we're all going to be looking at and evaluating into the future. The great benefit too is these are antimicrobial coatings, not a replacement for terminal cleaning or for disinfecting daily disinfection of surfaces, but they certainly are a benefit to help, especially with this biofilm regrowth. And they're a passive method. So you apply these in occupied rooms, and they are continuously supplementing your manual cleaning protocols. So I'm just listing here some of the methods. If you're interested, I've listed the publication where we published all of this data in a lot more detail, but just to let you know that our testing was done to the Japanese industrial standard. Remember, we don't have a standard in the US, but we adapted this method to evaluate these NADCC tablets and applied them to hard non-porous surfaces, allowed to air dry. I think I have a schematic here that tells that a little bit better. Air dries for 30 minutes, and then we apply our concentration of bacteria and evaluate what happens to any recontamination events over time. And we evaluated it at short time periods and then up to 24 hours. And these are the results with C. difficile. So this is, kind of the holy grail. If you can get rid of C. difficile, A lot of the other pathogens are a lot easier to control. So what you're seeing here is at the different time zones, and I want to just jump right down to the bottom at 24 hours, when we seeded with our bacteria after 24 hours of the surface coating being present, we found almost a 5 log reduction of the organisms. There were no organisms still present. We reached our limit of detection at the higher end. So that's a 99.998% reduction. And so our goal is to increase that original inoculum so we can show an even higher efficacy, because what this is showing is that the product is achieving even higher results, but we didn't have a high enough seed to evaluate that. So that's something that we're working on from the research side of things. Similarly, when we look at Staph aureus contamination, after 24 hours, we again hit the limit of detection at over almost a 5 log reduction, 4.9 log reduction, hitting our limit of detection. So highly, highly effective product. At all of our timeframes, we achieved the maximum efficacy of the product that we were able to test. So what can we sort of summarize from these results? We did the testing with a modified protocol. We're hoping the EPA is going to come out with some standard protocols so that we can really start to evaluate multiple products on the same playing field, essentially. And the results indicate that both Staph aureus and C. difficile, when coming into contact with this undisturbed treated dry surface, we see a dramatic reduction in cells, and they do not recover even after 24 hours. So our research, other research in biofilm prevention, this research was done on Montana State University. And so they provided this data to me so that we could work to summarize the data together. And so this was an academic focused project. MSU is well known for their studies in biofilms in multiple environments. And so they evaluated the NADC tablets, and they looked at what happened when the product was applied to steel coupons, air dried, and then adding Staph aureus bacteria and growing it, incubating it in conditions that would promote biofilm growth. And then what they did was both cultured what was left on the surface and then also did the backlight staining so we could see the difference between live and dead cells and any residual cells that were still present. Okay, so what we see here is that the control was seeded with over 5 logs of the bacteria. We applied the NADCC, and ultimately what we found, very consistent with some of the previous results on surfaces, is that even in a biofilm environment, we were achieving our upper limit of detection, a greater than 4 log reduction of the bacteria. So these results indicate that an undisturbed surface with a biofilm promoted growth, the NADCC product prevented the formation of biofilm over a 24-hour time frame. So I think this picture is a little bit harder to see, but on the left you see primarily green cells, and on the right, after treatment with NADC, primarily red cells, and a much higher reduction than what we saw previously with just bleach and the planktonic cells. So this is in a biofilm environment, so high efficacy in the most difficult environments. So this is more data, and this is from Medintech, the manufacturer of NADC tablets, of the effervescent tablets, KK2 tablets themselves. And so this is not data that came from any of the labs that I evaluated, but this shows the progression of the experiments, more replicates, higher concentrations of the initial seed environment. And what they're reporting is up to over 8 logs and almost 8 logs with Staph aureus log reduction using this product. So even just a 4-minute treatment of NADC with an added surfactant is showing really high efficacy. So stay tuned. There's more data that's going to be coming out, more publications evaluating this particular product and other CADs. And I think there's great hope that we can have better control in these biofilm, dry biofilm environments. So just a little summary here of some of our pathogen survival times that have been measured on dry surfaces. And, we're seeing some pathogens can survive for months to years. And, this is something we really need to get a handle on. And, I think these CADs are going to have a big impact on these endemic rates of infection that, seem to have evaded us. I mean, we have people who, systems and hospitals with incredibly stringent protocols and, incredibly effective, show efficacy and how these protocols are applied, and we can no longer measure any residual bacteria. But again, we're looking at that planktonic bacteria, not these biofilm embedded bacteria. So we need a new protocol on how to measure our efficacy, and then how to really get at this additional layer of dry biofilm exposure potentials. So my last couple of slides here, I just want to end on the CDC's guidance for best practices for environmental cleaning in healthcare facilities. And we are all familiar with what is recommended, the ideal properties for a disinfectant that's used in healthcare for environmental cleaning. Ideally, we want it to be non-toxic. We want to be able to use it when the patients are in the room, and it should be easy to use for environmental service personnel. If it's difficult to use or dilute, if it's not shelf stable, these are all things that decrease compliance and decrease efficacy. So, odors are another big problem. There's, certain disinfectants that are just not pleasant when you smell it, and they smell toxic, and people just don't like them to be used, even if they're effective. So, these are some of the things that we've been struggling with and trying to develop products to address these ideal properties. But the thing we really haven't talked about addressing are these dry biofilms and the need for that level of efficacy. So, again, more additional ideal properties for disinfectants. They should be broad spectrum, and they should act quickly. We want to make sure that the contact time is feasible for when we're cleaning and how long surfaces stay wet. I live in Tucson, Arizona. It's an incredibly dry climate. And so trying to disinfect surfaces where there's very low humidity, both indoor and outdoors, can be a challenge. Keeping a longer contact time, things to remain wet, it can be a very big challenge in some environments. So, and, environmental factors have a big impact on these efficacy rates and some of the difficulty of achieving the level of efficacy that we expect from these products. And material compatibility, corrosion is a big problem. We want to make sure that these products ideally are compatible with a wide variety of surfaces. So, but ultimately here, what we're talking about, a key issue is the persistence of these bacteria and dry biofilms. And we want to make sure we have residual antimicrobial effects on any of these treated surfaces to achieve, that goal. So that's the end of my slides there. I think we have a little bit of time, thankfully, for some questions.
Thank you so much, Kelly. That was a really, really good presentation. We have a couple questions. If you haven't, if you could, there's a Q&A icon. We had one question about fogging. I don't know if, Kelly, you want to answer. I know from my view, there's some pretty strict guidance from the EPA on fogging in terms of PPE and time needed after fogging. But I didn't know if you had any feedback on that question.
Sure. So we've done a lot of research on foggers, and I think one of the big concerns with the EPA is that people are buying fogging machines and putting any disinfectant in them, and so that's incredibly dangerous. There's very few products that are actually rated by the EPA as safe to be used in fogging machines. So that's the first key point. And even still, just the aerosols that are created by the foggers can be pretty intensive irritants to the lung. And so a lot of our research is done with firefighter populations who have already, because of the nature of the jobs they do, irritated lung tissue. And so the foggers can be even more irritating. So you don't really know who you're exposing when you're doing the fogging. So incredibly important to make sure whatever environment is fogged, if you're even using the right products and fogging machines, because we see a lot of people using do-it-yourself machines as well that they bought from paint stores or what have you. And this is, again, very dangerous. So, people who are asthmatic, they can also have a reaction to the foggers. So the aerosols. So you really want to make sure that the environment is cleared after fogging. And once you take the time to do those steps, there might be a better method to use in terms of, liquid surface disinfection. But yeah, so that's a great question. A lot of people want to know, can we just fog the whole room and be done with it? But there's a lot of nuances to that.
Great. There's a question, I think it's a good one. Is there evidence that biofilms lead to HAIs?
Yeah, so definitely the biofilms can lead to HAIs and the contribution they can make to the environment. You can look at source tracking genetically and see the same type of organisms, but how they're released, I think, the real point here is, how are they released into the environment so that people can be exposed, either transferred to the hand from the touch or aerosolization when people are in the environment. And so I think there's a lot we don't know about that and the real risk assessment, the exposure potential from the biofilms, but we know that these biofilms are growing in the environment and they have the potential to break off and expose patient and staff population. So I think, some of that data remains to be seen, but where there's pathogens present in the environment, there's always the potential risk of exposure, infection, illness, and then the more adverse outcomes.
Yeah, I would totally agree with Kelly on that question. Where there's smoke, there's fire. And I would certainly assume that because of these biofilms' ability to persist and break away, that they certainly could lead to HAIs, even from the standpoint of an opportunistic type of bug and not a resistant one.
Also on the topic of biofilm, the question is, if bacteria are so stuck in the biofilm, how do they break loose to be transmitted?
Yeah, so I can take that. Again, these are some of the things that we just don't really know. A lot of our biofilm research studies have been from the water environment. And so when you look at the water environment, same thing, you start with a biofilm that's heavily attached, strongly attached to the surfaces of the drinking water distribution system. But as they grow, they become more fragile. If you think of Dr. Rohde's visual where there's the attached biofilm that's probably very difficult to get off, but as it grows over time, you saw in his visual that it kind of raises into this mountain effect, and then any kind of shearing can break that off. And so what happens in the water treatment industry is you get these big chunks of biofilm breaking off once they get big enough to be vulnerable to that. And I think the same thing likely happens on surfaces. Once the biofilm gets extended far enough above the surface, it becomes more vulnerable to, if you're touching it, to it breaking off. And now you're being exposed not to a few planktonic bacteria, but a chunk of bacteria, still microscopic, but it's on your hands, and there could be a much higher concentration present. So controlling it at the lower level, keeping it at low concentrations so that it is a biofilm that's heavily attached to the surface or is not present at all is really our goal here.
Yeah, and really great questions in the Q&A. And Kelly touched on this. I think a lot of these questions literally lead into the types of research questions that we have. So some of this just isn't known. And again, I'm going to keep stating that you want to assume there's problems where there's smoke, there's fire, because as they break off in these bio kind of films that have higher, not only higher populations of these resistant bacteria, but also longer dwell times on surfaces, all of that leads to a higher risk. So I think the assumption can be that these are problematic, and certainly we need to do the research to bear that out.
Great. Just a couple more. We're not going to get to all the questions, but we have recorded the questions, and we will get with Kelly and Rodney and elsewhere if we need to follow up within the next couple days. But can you comment on some of the new technology that claims antimicrobial efficacy for 30, 60, or even 90 days?
Yeah, so again, I think there's a wide variety of products out there, and really comparing them and making sure they've been evaluated on the same standard scale is a challenge right now. So, I would have to look specifically at data on what kind of products you were talking about, how they were measured, what kind of standard they were evaluated against. But I think that's our goal, to look at the development of these products. How often does a surface need to be retreated to maintain the efficacy? There's a lot of variables that need to be evaluated. So your question is really a loaded question that I think I probably can't answer quickly here without some more information.
Agreed.
I would like to, I do see a comment in the chat, and something that I failed to mention in my presentation, that when I showed data on C. difficile, that was a stock of spores. So we did culture and culture the vegetative cells, but in the experiment on the surface and in the biofilm, they were spores.
Okay. The last question, I guess maybe for Rodney, how do you think SARS-CoV-2 will impact multi-drug resistance moving forward? I've seen some articles on this.
Yeah, great question. I think it's one being asked and studied presently. So if you look at some of the data that's popping up, there are studies looking at these types of issues, because remember, in the population of hospitalized patients, so if we look at healthcare and patients that are on ventilators and on other breathing treatments, and maybe even other types of interventions with IVs and things like that, we are seeing, unfortunately, that there's a higher prescription rate with antibiotics. Because what's happening, again, this is kind of a long soapbox thing for me that I can't get on, but when you're treating empirically because you think you might have something going on with pneumonia, for example, you may be treating in a sense and selective pressuring for resistant pathogens while you're trying to deal with something. So for something like a virus like SARS that's creating respiratory problems like pneumonia, you can kind of see the link there of why physicians might want to treat with antibiotics just in an empirical way. And we have to be careful and continue to watch this and interact with healthcare in a way that makes sure, we certainly know we have to treat empirically at times, but we don't have to do it every time. We need to make sure we're looking at laboratory confirmation of what's going on and trying to reduce those antibiotic prescriptions and usage so that we cut down on that selective pressure of antibiotic resistance pathogens. So I think it is there, and I think it's happening, and I think we're going to see the outcomes of that as we go forward, that there might be an increase in some antibiotic resistance problems in the next year or two, unfortunately.
Excellent. Well, thanks for everyone's time. Kelly and Rodney, you both did a great job with your presentations. We didn't get to all the questions, but like I said, we recorded them. Look for an email from TIPS if you're interested in the CE credit, and TIPS will also be hosting some additional webinars in the next coming weeks and months. And everyone have a great day.
Thanks, everyone.
Thanks, everybody.